Daniel is learning something new.
Oh, Daddy saw your painting. Daddy's black magic will surprise you. Place this order for my son.
Order confirmed.
I think we got it, the life we wanted. It feels like everything is
Sleep, baby.
going our way.
Wow.
Mother Earth, I'm connecting to the future.
Are you ready? Ready.
Let's start.
We are live.
Hello, world.
Days as if we just take it. There's no reason we should waste it. There's no miss. Ready now for all that's coming. We're standing at the edge of all that lies ahead. We got it, the life we wanted. It feels like everything is going our way. I think we made it after all the waiting. It feels like everything is going our way. We put every doubt behind
Daniel,
us. All the hope we hold
what did you see?
defines us.
Mama, a bigger fish.
It's called narwhal.
Tomorrow and every single day.
Honey, Daniel is learning something new.
Oh, Daddy saw your painting. Daddy's black magic will surprise you. Place this order for my son.
Order confirmed.
Good afternoon, ladies and gentlemen. Welcome to the Huawei Cloud Live Expert Talks. My name is Alex. I'll be your moderator for today. Please welcome our speaker, Wu Xing, CTO of Grandomics Biosciences. He's going to explain how they combine genetic sequencing technology and cloud computing technology to control the COVID-19 pandemic. Welcome, Mr. Wu.
Hello, everybody. My name is Xin Wu, and I am the CTO of Grandomics, one company focused on the third-generation sequencing services and solutions with Oxford Nanopore platforms in China. Today, I would like to talk about our distributed real-time mobile pathogen genomics surveillance system, which we developed during the worldwide epidemic of new coronavirus 19. You may wonder why the name of the system is so long. Well, I would say I couldn't find a compact name for it, since every word in the name really matters in reality, and I will tell you the importance of each word in my talk. So just keep in mind of these words: distributed, real-time, mobile, and genomics. So let's get started. So this is a dashboard of COVID-19 disease developed by Johns Hopkins University. It tracks all of the numbers of this epidemic. I made this screenshot a week ago, and actually the numbers shown here are outdated. The latest number of total confirmed should be about 2.7 million, and the total deaths should be about 190,000. So over 36% and a 49% increase in 1 week, respectively. That's really a disaster. We must take it seriously and find other ways to beat this virus. We must gain more knowledge about the virus itself, such as its structure, genomic information, etc., because they determine the way it infects human beings and its virulence. So let's take a look at what it is. The upper left is the electron microscope picture of new coronavirus 19 shot by NIH. You can see there are many specks on the outer edge of the virus particles. With specks on the outer edge, the virus looks like a crown. That's why we call it coronavirus. The picture on the right is a more precise illustration of the structure of the coronavirus 19. Basically speaking, the coronavirus 19 is a positive single-strand RNA virus. You don't need to know about the details, but some facts. One is the yellow helix shown inside the virus particle is the genome of the virus, made of RNA rather than DNA. The virus can make proteins according to the genes located in the genome, including proteins which make the ball-like virus particle and the spec proteins in pink color in this picture on the outer edge of the particle. Those proteins are the keys of how virus functions. As we said, genes are located in the RNA genome, and the RNA, put it simple, like a string, made of 4 letters, A, C, G, and U. The bottom picture shows the whole RNA genome of the coronavirus 19 is composed of 30,000 letters and contains about 10 genes, as far as we know. The key point here, there are many coronaviruses other than coronavirus 19 existed in the world for a long time, and the spike gene is most common component of the coronavirus genomes. It's also the most variable part. More importantly, the spike protein can combine the receptors of the host cells, such as ACE2, and play the key role to let virus enter the cells, as we call it infection. Okay, it's a little bit complicated, but don't worry, we will go to the easy part, hopefully. So maybe you get it now and have some questions in your mind, such as, okay, we know more about it. How does coronavirus testing work? Let's get into it. There are many products or kits available so far for coronavirus 19 testing. Basically speaking, they can be grouped into 2 types or mechanisms under the hood. One is by protein reaction, particular antibody, and the other is by DNA reaction. Let's talk about former first. When the pathogen infects human beings, the immune system of human beings will make kinds of antibodies, such as IgM and IgG, to fight against the pathogens after infections. As illustrated in the picture in the right hand, it might take a couple of days to make it testable. Antibodies testing itself is most fastest, cheapest, and the easiest way to operate. It's just as same as a pregnancy test strip. Without any testing equipment, you can get the result in minutes by the patterns of the bands. So if you see testing product claims it can do coronavirus testing in 5 minutes, without any extra machine is most likely to be antibody testing. The drawback of this type of testing is it does not help to stop spreading, since it cannot test the early stage infection, and is less sensitive than DNA reaction, which could be further grouped into two subtypes: quantitative RT-PCR and the DNA sequencing. Quantitative RT-PCR method is actually the gold standard for the coronavirus 19 testing around the world. I'm not going into the technical details here, but the basic theory behind the scene is that it targets 2 or 3 positions of the whole virus genome. We can make probes specific to these positions by exact reverse matching and combine to this size with the fluorescent markers, which are deactive under such condition. If there are some coronavirus 19, quantitative RT-PCR will make enrichment, and at the extension step, the probes will be cut and the fluorescent markers are activated and captured by the machine. After the operation, if the signal is above the threshold, the result is positive. Quantitative RT-PCR has a good balance of the sensitivity and the specificity, timing, and cost. It can test early stage of infection if nothing's wrong with the sample collection. So usually it requires lab work and equipment and take hours to get the result, and it's more expensive than antibody testing. Well, even gold standard method has drawbacks. Both antibody testing and the quantitative RT-PCR cannot provide coronavirus 19 genomic information, which is, if not more, at least as same as important of testing of positive or negative, yes or no. Why? Think about it. How can we design the probes for the quantitative RT-PCR without genomic information? And what happens if letters in these 2 or 3 positions change over time? How can we develop drugs or vaccines without genomic information and without monitoring changes of the targets? How can we know about the subtypes and the variant of the coronavirus 19 and the corresponding prevalence regions without various genomic information? We cannot give an answer. So here comes the DNA or genome sequencing. The more we know about the virus genomes, including its subtypes and the variants, the sooner we can stop the epidemic, the better we can make treatments to the patients. With the genomic information, we can absolutely answer lots of questions based on the similarities between different coronavirus genomes. Here is a comparison of the 15 genomes. So we can tell that coronavirus 19 is in human beta coronavirus group, more closer to SARS, but definitely not same, since they are divided into 2 clusters, and more far away from MERS than SARS. One thing should be noted is that even they look like so close or similar to each other, actually based on the difference between them and the molecular clock theory, they are still far away from evolution biology's viewpoint and not possible to derive from SARS or engineered in lab. As we said, the length of a coronavirus genome is about 30,000. So let's zoom in a little bit and focus on the Spec gene. The Spec protein has 2 subdomains. We call them S1 and S2 respectively. S1 has a receptor binding domain as illustrated by picture 1. Picture A, please notice there are 7 sites mutated in this binding region, and they are believed to be reason why it can bind human receptor ACE2 and infect human being. The ACE2 domain is believed to play a key role in membrane infusion of coronavirus to human cell. And one more thing, there is new insertion between the boundary of S1 and S2. It can improve the efficiency of the cutoff S1 and S2 domains, and is believed to be helpful to the infection. What we emphasize there is the genomic information really matters in our understanding of all of the pathogens, not only to coronavirus 19, all of the downstream activities like drug and vaccine developments depends on and benefits from these knowledges. Besides the mutations and the variants, there is also another interesting thing related to the coronavirus genome. It's subgenomes. The subgenome is a collection of combinations of genes or blocks along with the genome. Virus proteins are actually made from these subgenomes. It is still not clear what role it plays in the life cycle or the replication of the virus, but definitely it plays an important role in interaction of proteins and assembly of the real-life virus, which can infect human beings. The interesting thing we found was there are some dead or deactive subgenomes RNA can compete with the live or active RNA to function correctly. We could leverage the knowledge of subgenomes to develop vaccines by different strategies. Here we can make a conclusion. The more whole genomes, the better we can do to fight against CGU. Blogs in different published to discuss the mutations of the genome of coronavirus 19 based on two mutations located in position of 8,782 and 28,144. Some scientists grouped coronavirus 19 to two strains, L-type and S-type, and made some hypotheses such as L strain derives from S strain, and L strain might have more capability of spreading. This hypothesis is under argument of scientist communities, but no doubt with more genomic information we can approach to the truth step by step. Unfortunately, despite of 2.7 million of people are tested positive, nowadays we just got probably less than 4,000 whole genomes of coronavirus 19. So it's only about 0.15%. What we learned from these 4,000 genomes, we know that we found 20 point mutations, which is not that bad. And with some extra metadata, we can do some genomic epidemiological studies, such as the dominant strains in some regions and the spreading patterns around the world. The mutations distribution in the genome and the potential effects of these observed mutations. If you want more detailed information, you can visit the nextstrain.org, which is, I think, the best source of coronavirus 19 genomes. Another question might in everyone's mind is that where is the patient zero? Now, when did the epidemic happen? Maybe we can never answer the former question, but with enough genomic information, we can deduce the later. Here comes the epidemic dynamics. Based on the current genomic information, we can estimate there is about 27 letters mutated per year, and the beginning date of the epidemic is about middle of the December of 2019. There might not be so precise, but the trend tells us if you look at the black line, it's very similar to the regression analysis. You can get the same conclusion. Okay, so far we spent a lot of time on the biological science behind the scene. It sounds like getting off the track, but it helps us better understand the importance of what we did and what we will do. So let's get down to the business. We know that we need to sequence the coronavirus genomes if possible when we make a testing or screening. Here is the current paradigm to do the testing. No matter quantitative RT-PCR or genome sequencing, and no matter where it's operated, in hospitals or testing centers, they are designed inherently centralized. It brings a huge problem: contamination and cross-infection. Infected people and healthy people are inevitably crowded in the hospitals, even they can be specific for the virus testing only. We have no choice, and it's difficult to make a change since the buildings are fixed and the equipments are not portable. And we cannot make it flexible and scalable either. But with new technologies, we can redefine the paradigm of not only the coronavirus 19 testing, but also a more broader scenario. All pathogen genomic surveillance, followed by the words I can now make a more compact title: distributed, real-time, mobile. If we can put a lab, including DNA sequencer, inside a car or van, and use wireless network to transfer data in real time to the cloud and make analysis in the scale, we can make testing distributed, such as the big data analysis paradigm. Move the computing to the data, not vice versa. Move the testing capability to the field, not vice versa. And at the same time, mobile sequencing lab with 2 to 3 persons is inherently scalable. We can send many teams to many regions on demand and simultaneously. In this way, like Hollywood way of don't call me, I call you, the testing reach out to the targets, and it can avoid contaminations and cross-infection as much as possible. Meanwhile, it's much more flexible and scalable and can provide quickest response to the pandemic. So, a perfect new paradigm, right? So let's get to the details. We use most portable Nanopore sequencer, which is the 3rd generation single molecular sequencing technology. The smallest sequencer is called Minia. It's only a USB-C device. The bigger one is called Gradient. It can support 5 flow cells simultaneously. Its size is only like a PC. With nanopore sequencers, the DNA can pass through the nanopores on the flow cells, and the electronic signal can be transformed to DNA sequence in real time, which means if we test enough virus DNA, we are good. We can stop sequencing anytime. This is the fastest and the most portable sequencer technology around the world. Next, we design a mobile sequencing lab. There's not much magic behind the scene. Just made a modification of a vine to be a lab. Find all equipments which are portable and can be put into the vine, and bring 5G wireless device which supports tens of megabytes uploading speed in our testing. Then we developed a coronavirus 19 whole genome kit according to the RT network protocol to allow us to capture all the virus transcript DNA from the sample and integrate the Rampart monitor tool to our system to allow us monitor sequencing in real time. We are so grateful to RT network for the open source protocols and the tools. Their contribution is invaluable to the world wide. Before we can get started, we developed a grand pathogen system based on open source Galaxy platform. With the integration of the Rampart, we can support multiple sequencers at same times and can monitor over 100 concurrent samples, 24 samples per flow cell, and 5 million sequencers. Then we can get results of positive or negative for samples. The bottom left picture shows 2 positive samples here. You can see the coverage of the virus genome is about 100%, and the depth of the coverage. Some regions are relatively low because of the primary efficiency. Now we are able to get a better result with version 3 of the RT network protocol. The other highlights of our grand pathogen are: we provide a variant discovery pipeline which can tell us the high quality variants. And we double check a lot of samples, and they have a perfect agreement to our validations. We also integrate genome visualization tool that can help users to see the variant and their annotations in different regions of the genome. The upper right picture shows 2 variants indicated by blue and red vertical lines with the annotations. We also provide a phylogenomics pipeline, and it can construct a genetic tree for the evolution analysis based on the mutations. Grand Pathogen is a comprehensive and general platform for all of the pathogen analysis. Besides the coronavirus 19, it supports metagenomics pipeline for unknown infections and antimicrobial resistance pipeline for treatment decision making. Finally, the Grand Pathogen can fully leverage the Huawei 5G and the Huawei Cloud for data transfer and pipeline analysis. Under the hood, it incorporates the bioinformatics engine, Kernel, which is the implementation of Kernel and developed by us and Huawei, running on the Volcano batch system of Kubernetes. This kind of design made the whole system faster, easier, and more efficient than ever. Until today, we have sequenced more than 500 of coronavirus genomes, and we are fighting against the epidemic as hard as we can. As a human being, we never give in. That's all for today. Greetings to all frontliners in fight of coronavirus 19. Thank you for your attention.
All right, thank you, Mr. Wu. Thanks for your insightful analysis and thoughts. I think people around the world would really appreciate your efforts. And here we go. We've got many questions from our viewers online. Mr. Wu, your turn again.
Okay, we have 3 questions here. So the first question is, in the clinical testing market, will the market share of the 3rd generation sequencing usher in explosive growth? Yeah, it's for sure. The question is when. If you look at the current market of NGS, you will find all the sequencer companies directly or indirectly make investment to the 3rd generation sequencing. And I believe in 5 years, whole genome sequencing will be the routine work in the hospital. same for the third generation sequencing, since they have huge advantages of the chromosome and the genome level capability. If the more you can see, the more you can benefit, then it's just a matter of time. Stay tuned. So the second question is, why does analysis platform on the cloud use Kubernetes instead of virtual machines? Okay, I think long story short, in my humble opinion, the war between container and the virtual machine has been over more than years. And the war of container orchestration between different technologies has been over for a few years already. No doubt, Docker and Kubernetes are the winners. Kubernetes with a container can provide most flexible and scalable way for large computing world, especially for the biocomputing world. As we know, recently Broad Institute, which is the largest sequencing institute around the world, published a book called Genomics in the Cloud. I totally agree, with the infrastructure design idea in this book. And this is also what I did in the past years and what I will do better in the future. Regarding to more than 8,000 bioinformatics tools available to be integrated, you will be mad about it if you don't have enough lessons. So I think the current paradigm of the trend of the genomics platform development is a good answer for this kind of question. So let's go to the last question. What's the prospect of AI and the big data in the gene sequencing field? Actually, it's already there. The only thing is you are not aware of it. The better way for prospecting is to search the keywords of AI and the big data and genomics in published papers, and especially for the jobs on the internet. I'm pretty sure you will be shocked by the number. Actually, I use the data generated by AI and big data every day.
All right, thank you, ladies and gentlemen. Thanks for joining us today. Thanks for showing interest. This brings today's Huawei Cloud Live to an end. In fact, Huawei Cloud is committed to working with all of our customers and partners all over the world to find innovative solutions to mitigate and minimize the impact of this virus. So please don't hesitate to contact us if needed. Thank you very much.
Thank you.